is an HHMI scholar and an amazing scientist, and has been a wonderful mentor to me. And you'll see a lot of work that we've done together. And actually, the first thing that we did was some work in humans, a lot of work in mice and flies since then, and more recently, been getting into the mass spec imaging part of things. So what I'm going to show you today is a lot of mass spec in humans and flies, and then I'm also going to try and incorporate some of our newest imaging work, because I think that's really where I think some of the future work is and where I'm really passionate about. All right, so for those of you that aren't in circadian biology, this is how I've learned about the field in the last 10 or 15 years. And it's thinking about evolution and thinking about the way that we as organisms have evolved in this context of an ever-changing environment. Right? The Earth rotates, the sun and food availability, all of these things change through the course of the day. And so we're entrained to do this. So in mammals, this happens through something called the suprachiasmatic nucleus in the brain. And there's a number of genes in mammals, a few of them are denoted down here, that regulate this and cause the cyclical daily cycles that we think about. In 2017, these three gentlemen, Mike Young, Michael Rosbach, and Jeff Hall, were acknowledged with the Nobel Prize for their work primarily in flies. And so they discovered this mechanism, or the molecular mechanism by which this occurs, through genetic screening in Drosophila. So this is important. Now, long before that, we'd known about this from a perspective of metabolism. So this was one of the first papers I could find on metabolism and the circadian clock. And this goes back to 1928, where they reported that the ratio of bile acid to glycogen changed diurnally in I think this is in rabbits, right? So the amount, or the ratio of these two compounds is changing constantly through the day. And this period, like 2010 to 2015, 2016, there was this real interest in metabolism and circadian metabolism. So this was a nice review of that time by the late Steve Brown, who described some of the important factors by which the circadian clock interacts with metabolism. So it's things that you would think about, things like energy, right? Because we have food availability or we have activity, and these things all change. But then it's also things that are a little less obvious, like cellular translation or xenobiotic metabolism. So these are all regulated by proteins, or a number of them are regulated by proteins in the clock system. So one of the first studies we did with mammals was with John Hoganish, who's now in Cincinnati, and we looked at a number of compounds, and so we used an targeted ion switching method, which we developed, and we measured in a very high resolution every hour over 48 hours the levels of a number of metabolites. And what we found was about 50% of what we could measure was actually changing in terms of the circadian cycle. And so to me this is very provocative, and it still is, because it says that if you're looking at something in the liver, and I think that this extends to many other organs as well, if you're looking at something in the liver, it depends on what time of day you're looking at these things, and you're going to get slightly different results. And this was noted by others in other fields, in the transcriptional world, a little bit before. But this was really the first strong demonstration of this in mammalian tissues. So around the same time, we had started a project in humans, and this was before my lab was actually set up. And I'd sent the samples to Oliver because I knew about his work and trusted him to be the source of our metabolomics data. And so what we found in this case was related to sleep, on the sleep side. And this was that if we sleep restrict individuals for 5 days, we measure them at the baseline, we measure them after 5 days of sleep restriction, we see pretty dramatic changes. And then in most of the cases, you see that there's a recovery of those metabolites. So they come back to a normalized baseline value. But in some cases, they don't. And the things that don't are things like carnitines. 4-PYR is a nicotinamide breakdown product. And so these are things that are going to come back through this talk, some of these things. So even though this was early data from humans, I think it really sets a nice foundation for what we found in the flies later on. And so what we took away from that particular study was that if we take these effects of sleep deprivation, So there's neurological, neuropsychological defects, there's immune defects, there's metabolic defects, reaction time things, that we can sort of turn that into a view of metabolism, right? We can say, okay, well, what are the specific factors that relate to each of those factors? And what we've done in the last few years is try and understand a little bit more about some of these. And there's obviously lots to study, but this is the sort of foundation from which we started a lot of our fly work. All right, so why flies, and are flies useful for this? Obviously, and having dedicated like 10 years working on this, I think so. So hopefully I can convince you guys as well. So flies meet the general criteria for what we consider sleep, meaning that when they're asleep, you have to poke and prod them more than when they're awake to get a reaction. So they have this arousal threshold. That arousal is different from if they're under some kind of anesthetic or coma. The sleep that they have is under both circadian and homeostatic regulation, meaning there's a time of day effect, that's a circadian effect, and there's also a homeostatic effect. So if you sleep deprive them, the sleep pressure increases more and more. And then others have shown all sorts of neuronal activity changes. So these are important features of sleep across all mammals and other animals, and they're also observed in flies. In terms of circadian studies, I talked a little bit about the mammalian clock, which I'm not going to talk about too much today, but this is kind of the core clock design in mammals. And Drosophila, and as I mentioned, was the first real clock to be discovered in a higher organism, and you'll see that it has a very similar layout. And one of our first fly studies, the first data we really published out of our lab, was showing that there is this variation in flies as well. Right, so in this particular case, I'm looking at just a regular wild-type fly, and we're just showing that there's variations. And what we showed in this particular paper, which I don't have the data for here, is that if you knock out the clock using something called a PER0 mutation, so PER is this protein right here, that you basically get very little cycling. And so this is dependent on having an intact clock in flies as well. All right, so throughout the talk today, I'm going to refer back to two mutants, Fumin and Sleepless. So I'll just describe a little bit about them. Fumin has a mutation in the dopamine transporter gene. So if you think of the synapse, dopamine usually gets taken back into the nerve cell. But what happens in this particular mutant is you have a buildup of that dopamine, and so you get increased dopamine transmission. And so in these dopamine mutants, they have reduced sleep, they have increased activity, and in the original paper for this, it was shown that the lifespan was the same. Now in our hands and in our background that we're using, we actually see slightly different lifespan. But more or less, it's still much longer than most other mutants. So the mutation is really not that detrimental to the fly. And then we also see behavioral rhythms, as I'll show in a minute. Sleepless is a protein which impacts the membrane transmission, and what we see then is decreased GABA, and because of that we see increased activity and less sleep. These flies tend to be a little bit sicker, so they have a decreased lifespan, but they also show the same kind of behavioral rhythms. And so what do I mean by that? Here are activity traces. Normally I would have some confidence intervals on this, but in the whole transmission between computers they seem to have lost them. But these are very robust changes, they're very tight confidence intervals, and you see that between the wild type here you see a nice diurnal rhythm, and I would also normally have boxes showing the dark periods. So between 12 and 24 would be a darker box in the background, you can imagine, and 36 and 48 would be a darker box, and those are the nighttime periods essentially. And so in wild type flies what you would see is a transition at the dark day and night time periods. You'd see that these peaks in activity, right? So this is where a fly would be foraging, or before and after dawn and dusk. Now in Sleepless, you actually see a similar rhythm, but it is blunted, and you see that the total activity. So on this axis, activity per 30-minute bouts, total activity is much higher, right? So Sleepless has high activity. Fumin also has a high activity, but in this case, you don't see the second activity transition here, right? It doesn't peak. So at nighttime, which is where this would be here, you actually see increased activity through the entire night. You don't see this drop in activity as you do in the other two strains. So this is important, I think, to contextualize a lot of the other results that we're going to see. So when we start looking more in detail at, we're going to look just at human now. What I'm going to show here are some of the ratios and important metabolic features that we see from the two strains, right? So if we look at the glutathione ratio, NADP, NADPH ratio, that these are all cycling pretty strongly in the case of these two here in the ISO, the wild type. And so what I'm using is a tool called JTK, which assesses the cycling using a non-parametric test to look for basically a rank ordered change in data points through a time course. And then based on that, it assesses the fit and spits out p-values and q-values depending on how many tests you're doing. And so this is the negative log of the p-value here. And you can see that when I look between the wild type and the human, that there's a pretty dramatic drop, right? So that means that our cycling is basically gone. And you can see that visually in this particular, in this case here. This is the wild type, this is the human, and you can see the cycling here and no cycling here. So this is important. And so we reasoned that there's probably something interesting going on here. And what Danya did, who was a student at the time in my lab, Danya Malik, she took the two strains. So here's the sleepless here, here's the fumin here, and here's the wild type here. And she ran something called OPLSDA, which is orthogonal partial least squares discriminant analysis, which Oliver tells me now is considered a machine learning tool, which at the time I used it was a projection method, derivative of a PCA essentially. And along the first axis is where you see the most variation, the axis of primary variation. And you can see that along the first axis we see the difference between the wild type and the other two strains, which we'll call sleep loss strains here. And is this really? Yeah, I apologize for the resolution. I think as it transferred, I also lost some resolution to the figures. But the moral of the story is you see a pretty dramatic difference in overall metabolism between the sleep loss versus the wild type strains. When we started looking at what metabolites were underlying that, just by taking the sleep loss and then the wild type and doing enrichment analysis on the differential metabolites, one can see that by breaking it down into different phases of the day, that the primary changes are actually mainly in the evening, right? And when you look at that activity trace, that kind of makes sense, right? Particularly with the fumin, that the fumin shows more activity in the evening compared to the other two, and so it sort of makes sense, right? That the evening period is where you see the most activity change. Just as an example, and this was probably the top hit in the screen, was nicotinate and nicotinamide metabolism. When we look at the overall period, we see some change. When we look at the light only, we see some change. But if you break it down into early light and late light, there's no change that's significant. Whereas all of the dark points are changed. And when you look at the individual traces of nicotinamide as an example, you can see that come through. So I won't go through all the data because there's tons of it. But when you kind of put it all together, what we determined, or the takeaway message is that when we have normal sleep with a wild type fly, we have kind of this balanced reaction or balanced energetics. We see some changes in these nicotinamide and nicotinate pathways. In short sleep, that balance gets disrupted, and it makes sense again, right? Because we're now having a short in, let's say, these short sleep mutants, we have energy usage throughout the day, but more at night, and so the energy requirement is different, and it changes here. All right, so we have this nice steady state data showing these changes, but really what we're doing is we're taking snapshots at different times of day, and what we're missing is the dynamic information, right? So for those of you who have worked in metabolism, you may appreciate that one of the ways to get at this dynamic information is to use metabolic tools like metabolic flux, right? Where or metabolic tracing, where you introduce some kind of tracer, see where those end up, and then we'll do that. and then look at a bunch of data, and we will try to do some of that later. With flies, that's a little difficult because and in a circadian time course, it's extremely difficult because you have to introduce an amount of tracer that will be detectable, but also within a certain time period that you can measure, the actual flow of that, those tracers. So I'll show you one approach that we've taken towards the end of the talk. But before we get there, what we actually started with was something called respirometry, and this was what we set up. So this is a small animal respirometry system, and the rationale behind this is by using the indirect calorimetry, which is measuring both the CO2 and the O2 consumed, we can infer what the metabolic rate is of the entire organism, right? So now we're looking at the whole fly and saying, okay, well, how much oxygen are we consuming and how much are we CO2 are we evolving and oxygen consuming. And so what we measure as the primary output is something called the respiratory quotient. This is the RQ, and I'll refer to that a lot in the next few slides. And what that is the ratio, essentially, of the CO2, VCO2 over VO2. And the way we interpret that is that if it is near 1, that it's the primary source of fuel is carbohydrates. If it's well below 1, then it's primarily using fats, and if it's just below 1, it's proteins. So this is kind of the conventional way that calorimetry or indirect calorimetry has been done, particularly in mammals. These are where these numbers come from. And so this isn't something new to us. Alex Keen's lab has published on this a couple times and published a paper before we got into this, and that's how we kind of got into this. So we picked up this system, which is the small animal respirometry system, and it has different pieces, one to measure the oxygen, one to measure the carbon dioxide, and then a main unit that controls the flow. But because most people do this for other reasons and not for circadian studies, we had to come up with our own adaptation to make this work. And so we had to develop this dedicated enclosure, and so it had all sorts of mechanisms to make sure that we could control the lighting in particular, but also the temperature. And so this is a sort of nicely controlled box that we developed together with the machine shop at Penn, and has worked very nicely in our hands. And so some of the advantages of the respirometry are sensitivity. So if I want to measure carbon dioxide, I can do that from a single fly very reliably and with very good signal to noise. If I want to measure oxygen, it's a little bit noisier. So we use about 25 flies per tube in that case. But it's still relatively easy to do, and it's very high resolution sampling. So I can sample, every, I think, 5 seconds is what we're getting out of the instrument at the moment. And then, of course, we divide, or bin the data to get a little bit more reliable. So here's an example of 2 different experiments that have been averaged over a circadian time course in each of these strains. So this is the ISO wild type, this is the fumin, and this is the sleepless. An example of the data. You can see that you can see some circadian signals to this if you're looking closely. Or pretending, but I'll show you some data where we actually see that. And what we did is we looked at this in the strains I'm talking about, and also this per mutant that I talked about earlier, where we have a circadian clock mutant, just as a control to make sure that what we're seeing is not artifactual or environmental, right? That's really coming from the clock. And so when we take the measurements of all of these, and I'm not going to go through the CO2 or the O2, but let's just focus on the RQ here, the respiratory quotient. You see that in the wild type, in the ISO flies, we have basically the same relative signal if you average across the day as we do in the clock knockout or in the same wild type that is under a dark condition. That means that we're not changing the lighting at all. It's always dark during the time of measurement. Now for fumin, we see an appreciable drop in this, and then in sleepless we see an even more dramatic drop. Now, the question is, okay, well, what is the fuel source? So 0.9, so that would imply something like protein or lipid, according to this chart that I put up earlier. But we see an RQ greater than 1. And so when Alex Keen published his work in flies, he also showed the same thing, that their RQ values were all around 1.2. And his interpretation was that this is lipogenesis. And so we think that the flies are taking in, in their diets just, this rich amount of carbohydrates, and they have an excess, and then they're, converting it to lipids. So still not completely sure, of that whole metabolic picture, but that's kind of the working theory at the moment, at least for the wild type, and this per mutant. And then, as these, sleep mutants probably need more energy, and, are working differently, so that's why their RQs are different. If anybody has any thoughts, I'm happy to discuss that. So here are some of the measures of the phase. So this would be the peak time at which the RQ is peaking. RQ is in green, VCO2 is in orange, VO2 is in purple there. And you see that Fumin is not significant for RQ, or sorry, for VO2. But the only one that is not significant for RQ is the wild type LD, right? So the basic wild type condition has essentially a flat respiratory quotient through the day, whereas the other ones all have some kind of perturbation, and I think that's causing some time of day effect, right? And so I think what it shows us is that our wild type is really well and homeostatically controlled, and so it doesn't have these peaks and valleys, whereas the other ones do because of these perturbations. And I think that's a nice sensitive way. And in our hands, the respirometry, and Amita's been using this a lot, has shown that it's very sensitive to gross physiological changes, which is a really nice way of looking at this. So what we've done then is taken our metabolomics data, because now we have circadian time courses of all of these strains, and we have this very high resolution respirometry, and we asked, okay, well, we don't think that whatever we see in the metabolomics is exactly correlating with happening at respirometry, right? Because, again, the metabolomics is all a snapshot. It's all like a single time point. So if I'm seeing a change in, my O2 or my RQ, right, is that really when the metabolites are mobilized or when they're moving? And so what we did is this is something we're calling a lag analysis, right? And we took various time points. So this would be 0. This would be the metabolomics data lined up directly with the respirometry data. But then we also shifted the respirometry data by various time amounts, both positive and negative, right? So it's a little, tough to wrap your head around for a second, but let's say time 0 is here. We're going to move the respirometry data. Because it's high resolution, right, and we have it every 5 minutes, we can move it, anywhere from 5 minutes to wherever we want within the time course. And so by shifting the respirometry data, we were able to assess, well, is there a change in the metabolites preceding or following the change or the respiratory events, right? And so by doing this, here's an example for a lag of 120 minutes in quinolate. You see that there's a negative correlation in the rank, right? So this is a Spearman correlation. And on the other hand, if we do this advanced by 120 minutes, you see that or do I have that reversed? I might have that reversed. But you see that there's a positive relationship here between the hydroxy hexylcarnitine. And so when we look at the overall pattern across all of our metabolites, we found this set, which are either increasing or decreasing in a positive way before or after. And without getting into the weeds of it, what we say is that there's this sort of set of predictive metabolites, right? So these are largely energy predicting. So TCA cycle intermediates, carnitine derivatives, and NAD precursors, right? That are sort of peaking before, or this relationship is peaking before. It's not actually the level of the metabolite, right? It's the relationship to the respirometry. So it could be a positive correlation or negative correlation. It's not the level of the metabolite. Or we have a response metabolite, right? And so these are, when there's a respiratory event, these will be, changing as a function after the respiratory event. And so this is things like pentose and nucleotide intermediates. So what I think we have is a very, relatively sensitive assay, essentially, right, for what is changing in respiration. And so now we don't have it done yet, but we're working on the other mutants, and we have the data, but we have to do the analysis and figure out exactly, okay, so now when we change sleep or circadian rhythms, how does the relationship between the metabolites and the respiration itself change? And I think this is going to be pretty useful. All right. So the other interesting thing about Drosophila and challenging thing about Drosophila is the size, right? So as you can imagine, these are really tiny samples we're dealing with. And so for a long time, I've been struggling with, okay, well, how can we get tissue-specific data? And the reason for that is we know from transcriptomics and other studies folks like Amita have done using other tools that there is an important crosstalk between different tissues in Drosophila in terms of sleep, and in mammals in terms of sleep and circadian rhythms. And so, I've been thinking about this for a long time. And, there's all sort of sophisticated mass spec tools that you can use if you have the right sampling. But what we've kind of come to now is imaging mass spec as a way to understand tissue specificity. And I think we can do this in flies, but I think we can also do it in other organisms. And so I'm going to talk a little bit about the imaging mass spec as a way to deconvolve the contributions of different tissues. And this is something we've been spending a lot of time on in our lab. And so what we're doing is essentially collecting the samples, putting them on slides in a fresh frozen manner, collecting our data, and then we've developed a tool in-house that we're calling MSI Eagle to analyze this data. And because I know Oliver's group likes metabolomics tools, I thought I'd show a little bit about this. So basically the idea of this is to take your data in, and usually it's a raw IMZML file. And then what we're using is Cardinal, which is a mass spec imaging processing software, which is amazing and has a lot of really nice features, but it's a little bit difficult for non-specialist users to use because it's very command-driven, and there's really no interface. And so being in a translational lab, one of my challenges is having people come in with different backgrounds, biologists and people who are in math and in other areas, and some of them pick up the R programming really well, and some of them don't. And so what we decided to do is develop this sort of GUI, which would not only use the cardinal commands, but also add extra features to this. And so this is kind of the overall view of what the software does at the moment, and we're it's set up in a modular way, so we can add or remove modules as we see fit. But what we've been able to do with the flies, and this is some of the early data we collected, and I think this is at 20 micron resolution using our Waters DESI, is we're slicing them and then showing that there's a pretty dramatic difference in a number of metabolites depending on the specific area that you're looking at. So this is the different parts of the fly, and when we combine statistically the different regions that we have, and then we do a test, you see that there's differences both in the area that we're looking at. So obviously abdomen is quite different from thorax, from head, but within each area there's also quite a substantial difference. So here's an example in brain, and we see different metabolites. And when we look a little bit deeper in some of those metabolites, when we compare that to gut, you can see what we're seeing are changes in the ratios of a number of important energy compounds. So in this particular case, if you look at hexose and hexose bisphosphate, which we haven't, because we're doing the imaging, we haven't done the MSMS, but I'm pretty sure this is mainly glucose. If you look at the ratio here, you can see that the fumin is dramatically different in the brain, whereas it's kind of the opposite in the gut. And so, we're hopefully working towards getting this tissue specificity. And that's what we're kind of working on now. I don't have any real results to show you other than this preliminary data. But the other thing that we've been working on with our DESI source is trying to really get into some of the finer details of the data. And so this is 2 micron data, and I'm switching on you a little bit to mice just for a second. And so this is mice brain, which we collected at 2 microns, and so this is from the hippocampus. So the area that we collected was about 1 millimeter squared, as shown here. This is the neurons of the dentate gyrus that you can see here. This is H&E stained section, the same section that we collected on the mass spec, we then stained by H&E. And then we used a pathology software called QPath to pick the nuclei here. Use cell expansion, so I don't know if you can see this, but there's borders around the nuclei like this. And then we took those measurements from QPath, clustered them. So now we haven't, so far there's nothing to do with mass spec yet. We're clustering just based on nucleus and cytoplasmic characteristics of the staining. So that would be things like cell morphology, cell staining intensity. So there's about 20 different measures that we use and different ratios. Perform a cluster analysis, and we come up with 4 different clusters. And those are colored here. Then we apply that to the mass spec data. So we use a registration process where we look at the ions in the mass spec, determine where the cell borders are, and then apply those polygons onto the mass spec imaging data. And so now what we're getting, we'd like to think is cell-specific mass spec information for each of those clusters. And so here's an example of one ion, 788.54, which is clearly very different in this one cluster of cells. And when you look at the localization of those clusters, they look very biological, right? They're very real in terms of what they should be representing. And so if we take the mass spec data now and show that specific M over Z value, here is the areas in which that is highly enriched, which is in the neurons of the dentate gyrus. And when we overlay that with our initial polygons here, you can see that we are actually getting what I think is subcellular level resolution of what we're looking at. So that's kind of what we're trying to do, is really understand that. When we take our 4 clusters and then we look at the different compounds, this to me looks very real from a biological perspective, right? So we see high enrichment in things like acylcarnitines in what we think are the neurons, which is what you would expect, right? These are very energy heavy molecules, and then you can look at the other ones. And so we think we know what these clusters are, but our next step is to really go back and see if we can stain for specific markers of different cell types, and that's where we're at with this data. So coming back to flies, when we use the same kind of approach and the same resolution, we can take a fly brain, and these are the different regions that, so Pinky Kane is our fly biologist. Ooh, did I skip far? Pinky Kane is our fly biologist, and she understands these things much better than I do, but so she's drawn this nice sketch of what we expect to see. And these are 2 brains from 2 different flies. And when we do some kind of clustering, in this case UMAP, what we can see if you zoom in is that these are actually real clusters, right? So this isn't just noise. It looks like noise from here. But when you zoom in, you can see that what we're seeing are localized clusters. And I don't know if they correspond to cells or intracellular spaces, but they're definitely something that correlates to individual ions. And then when you look back globally, you can see that the 2 different areas correspond to the eyes and the brains within the fly. So I think using this level of detail, we can definitely come up with cellular resolution information, metabolism information in the fly. And in a perfect world, maybe we can even go down a bit lower. So I'm very excited about this, and this is something we're pursuing hotly. All right, so coming back to the big picture of the flies and what we're looking at is I've shown you all sorts of data and the respiration, the metabolomics, and what we're kind of coming to is this hypothesis that we have this rhythm of signaling, right, of normal daily life, and we have this energy rhythm. And in the wild type case, in a normal case, we have kind of a nice concerted balance between these 2. But when we have a perturbation, something like sleep loss, we're going to have a disconnect, and we're going to have some disharmony between these. And so the questions are really, how can we understand this? And so what I'm going to talk about in the next little part is glucose and glucose metabolism a little bit, and trying to understand how we can understand the relationship between these rhythms and the metabolic flexibility. So in normal life, right, we've evolved in this world which is, turning, and in the natural world, there's all these things that don't impinge on our normal everyday life, right? But in everyday life, we have all sorts of things that change the way that we behave relative to the way that we've evolved. And so there's this, sort of tension between how the body's evolved but what we do. And so, some people argue that this is leading to something called the circadian syndrome. And there's a broad range of, exposures that do this, things like light pollution, staying up late, social jet lag, stress, shift work. And we know that many of these things epidemiologically cause long-term metabolic consequences. And one of the things that we've known actually for quite a while, and I think the initial evidence was back from the 30s, is that there's a difference in glucose metabolism in humans through the day, right? So if you take in, and these are different ways of showing this, but I'll show you this one here. If you're taking in a continuous glucose infusion here, that you'll see a time of day difference. So this is at nighttime, right? And so you'll see that this is all plasma glucose in here. If you look at this glycogen concentration, so this is in controls, you see that there's a change. In basal and post-meal. And then in type 2 diabetes, there's really no change. And we know that this is this has a definite time of day effect. And so the question that we asked is, okay, so we know that glucose and glucose itself changes as a function of time, right? Glucose levels are very different in humans. And we're like, okay, well, what's the sort of broader glucose network? And we looked around and we didn't find anything really strong in terms of the metabolomics literature. And we had samples that were sampled every 4 hours. And so we decided to take this set of samples. So these are from individuals that have had 4 isocaloric meals spaced out over 4 hours. And then we selected 93 plasma metabolites that we felt were correlated to this glucose network from these 14 individuals. And what we found when we did the usual metabolomics and clustering is that we see a kind of a very strong network and very strong cycling. And so looking at this heat map, these are all cycling and then they're peaking at slightly different times, right? And then these are the different categories of compounds that are changing. So it's a broad range of compounds and a broad range of times, but this network is very tight, right? And so this was striking to me because when you look at some of the other literature where people have done work in humans and kind of a more broad-based selection or non-targeted screen of metabolites, like for example, Josh Gouldie's looked at lipids, you don't find the same kind of concerted behavior over all individuals. You see subpopulations and subclusters, but having this sort of really tightly regulated network is something unique in human studies as far as I know. And so, we thought this was interesting, and two kind of interesting sort of subpoints out of this is one, that this afternoon time point isn't really distinguished when you do a one versus all comparison of the different time points. You take one time point, you do a statistical test against all the others. Each point has its own kind of space, but ZT4 sort of sits in between everybody, and it's different. And so ZT4 would be kind of like right around noon, right? So it's just before the afternoon. And so, the takeaway from this is that we have this robust glucose network, and that we've measured the concentration of glucose in all of these clinical studies in the past, but I don't think glucose is really the only thing going on, right? There's obviously this wider network which constrains glucose in certain ways, and so I think that it's really worth investigating and understanding that. And then the other important point here is that the pathways that we're looking at are all very highly conserved, right? They're all things that you'll find in flies or in mice. So that's why another argument why I think we should be looking at flies, because it's a lot easier to study these things in these animals. So looking at the fly as a tool to understand glucose metabolism, insulin signaling, there's actually a lot of parallels between mammals and Drosophila in terms of what kind of signaling occurs. Right? So here's a table with some examples. And if we look at the way that the fly signals, is it has insulin, which is produced as an insulin-like peptide, which then interacts with an insulin receptor, and you get a number of the same compounds or the same signaling entities as you do in mammals. And so the general formation of the pathway is very, very similar. And so that's why we're working with that. And so I'll point out AKT, because that's one of the compounds that we're looking at in the next slide here. And then, I don't know if I have it on here, but we're also looking at PI3K signaling. Here it is, yeah, PI3K signaling in another set of samples here. All right, so what Pinky showed was by using whole bodies, so now she's doing Western blots of the bodies and looking at PAKT, she showed that in fumin, which is red, and iso, which is blue, that there are clearly time of day differences in the way that the insulin is being processed here. She then went on to use another tool which allows us to be a little bit more targeted and look specifically at cells. And so what I showed you here, this is the fat body, and these are the fat body cells where all of this is happening. And so she went in and is looking at specifically the PI3K, so PI3, K signaling in here, and then looking at DCAT as the control, and measuring within each of these cells. She's measuring this, and what's important in insulin signaling in this particular case is that there's a localization of a lot of these compounds to the membrane, right? So there's a shift from the cytoplasm to the membrane during the insulin signaling events. And there's also a number of redox processes going on, which I'm not going to talk about now, but I'll come back to at the end. And so when you take the ratio of the GFP at the membrane, so this is a measure of what's occurring at the membrane versus what's happening in the cytoplasm, you can see that in the fumin flies, right, we're also seeing this difference in the fat body as compared to the wild type, right? So now we can measure at the level of signaling that this is happening. All right, how much time do I have?
Don't worry.
Yeah, okay, so I'll talk in the last little bit then about a study which Danya started and we're just getting out now, which involves injecting flies with glucose. So I told you that this metabolic tracing is something that we thought was one way to be able to understand this. Now the problem with that, of course, is that you're now introducing some kind of tracer, and you have to do that in some way. But having come to this appreciation or feeling that the insulin signaling is really important, what we have done is actually basically been injecting the flies with trace amounts of glucose. And so what we do is we introduce the glucose with a dye, and then the dye tells us how much glucose is being injected by this tube, and then we measure the dye in the mass spec, so we can normalize for the amount of dye in our mass spec assay, and that tells us in this whole body how much we've actually got into the fly. And from that, we also test for physiological changes, so locomotion and geotaxis of the flies, and show that those are normal after an hour. And so basically what we're doing is essentially giving like a glucose tolerance test to these flies, and then we do this every 4 hours. So it's quite a big experiment. So we inject, and then we give 1 hour for the glucose to metabolize, and then we do the extraction and analysis by mass spec. I've kind of already talked a little bit about this, about why it's important to do the tracing, right? Because we're not looking at steady state phenomena. We really want to, show where things are going and how they're going, not just that they're there, right? And so this is the sort of set of metabolites that we looked at in this. And so for each of these, we measured individual isotopal logs using MSMS. So it's a slightly different method than what a lot of people use. But that allows us to, understand, at a very, high level, I think, or very, low level, depending how you look at it, what's happening to each particular metabolite. And then Seth Rhodes was a student who worked on this initially. He came up with this method for correction and calculating the metabolite enrichments. And as one would expect, right, we can confirm these changes in overall levels of these monosaccharides. We also see that there's an inverse relationship between the parent form and then, in this case, the N+6 form. And when we look at the overall changes in total for these, so these purple compounds are all the parent forms, and these are all the isotopolog forms. We see that the isotopologs cluster around ZT4, right? So our argument now is that we have, through this process, figured out that there's this biosynthetic kind of rush hour at ZT4 where you get incorporation of these of the glucose into all of these other compounds, primarily in the sort of morning phase or late morning in these flies, right? I hope everybody's following. Does that make sense? Sort of, yeah. And then if we look at fumin, we can do the same thing. And we see that the glucose fate is, we see two different time points, ZT0 and ZT12 in fumin that have similar points where it's very different. But when we look at what isotopologs are involved, only the one at ZT0 looks similar to the one at ZT4 in wild type, right? And so then the question is, okay, well, fumin has a very different rhythm, right? But when we line up the feeding, so this is an ARC feeding assay that Danya had done, you can see that the peak in feeding for both of these is very similar, right? It's only offset by about an hour and a half, and it's actually in the opposite direction. So where the fumin peak in sort of this rush hour was ZT0, its feeding peak is actually slightly later than the wild type. And so, to us, that suggests that it's really not feeding that's driving this. And also the fact that fumin has two peaks, right? Whereas the feeding peak is much earlier. We don't think that those, rush hour peaks are really related to feeding itself. Right. So now when we put all of this data together, and then we also did actually a fasted experiment where we fast the flies 4 hours before each time point. Then we see that the rhythm persists, right? So when we look at the isotopologs, so again, these are the N+0, and these are all of the isotopologs, that in fumin at ZT0 or wild type at ZT4 and wild type fasted, we see a very similar pattern, right? So that this incorporation of metabolites into the downstream pathways from glucose, we think is very robust, and we think it's under direct circadian control. All right. So, this is what we think from these injection assays, but I think now we need to go back and do some of the other work that we've done in the uninjected flies, right, to prove that what we're seeing is really happening. So that's kind of the next sort of set of experiments I think we need to do, is use this injection method, but then look at tissues and look at the other influences in there. So that's kind of where we're at with this. In the interest of time, I'll skip that. So this is our kind of final working model to put it all together. So the metabolic flexibility, we think is something that the flies are exhibiting, and that the sleep loss is clearly changing, as we saw from the respirometry data. And we're able to kind of measure this in real time using some of these tools, right? And so anything in red are metabolites that we're measuring or are cycling. So these are cyclical. So everything in the green boxes were measured, but anything in the in black is not necessarily cyclical, but we are measuring. But these are cyclical, these processes here. And so what we're trying to do is, put this all together, particularly with this injection assay, where we're able to, give them the glucose tolerance test and figure out whether this persists. And it's this sort of challenge, I think, which is really important to understand how the physiology responds, right, as we see from the respirometry. All right. So with that, I'll acknowledge the people involved here. So Arjun was really responsible for all of the human work. Pinky has done all of the most recent fly experiments. Farain has done all the respirometry. Danya and Seth set up this whole platform for the injections and the metabolic tracing, which is amazing. And Paula also helped with that. She was briefly shared between Amita and I for a while, and she helped with the respirometry and other things. Amita has been amazing as as I mentioned, as a mentor and somebody who's, been extremely helpful and, the real expert in the fly stuff to get us moving. And then these are some of the other folks that have been involved at some level in these projects. And thank you for your time and the invitation.
Questions for Aalim? Glucose metabolism. Did you guys test on the insulin resistance?
Test on insulin resistance.
Yeah.
So that is to some extent what we're trying to do with the AKT signaling, right, and the TPGH signaling. I'm not sure how to do that in another way in the flies. I don't know, Joanna, if you know.
There are movements that actually show similarity to insulin resistance phenotype.
Yeah. So, but we haven't, to understand, let's say, at different times of day, if a wild-type fly has increased resistance or not. I don't know. But I think that's what the insulin signaling should tell us, right?
Can you elaborate on the afternoon diabetes in human context? What is it?
What is it? Yeah, so if you take in, you do an oral glucose tolerance test at 8 a.m., and then if you do an oral glucose tolerance test at 4 p.m., right, you may switch from like a non-diabetic phenotype to a diabetic phenotype, right? So there is, it's a well-known phenomenon that and not everybody shows this, obviously, right? It's like averages across many individuals, but that there is sort of this change in the way that your body can take this in. And so if you look at our fly data, right, I showed this to Tom Jongans, who's also a fly guy, and he was like, so if I eat a chocolate bar in the morning, that means that my glucose is getting processed, in a certain way, and then if I take a chocolate bar in the afternoon, it's going to be, stuck somewhere else and probably go into storage as opposed to being used for catabolism, or being catabolized, right? And so that I think is a fundamental question, right? Is how does that apply? Do our results from this fly work really apply to other systems? And that's something else we'd like to show. Mm-hmm.
I really like the talk, thank you.
Thank you.
I just had a question about the time course experiment, which you feed glucose and you measure those metabolites. So is anybody thinking about doing like a flux balance analysis because you're generating this many MIDs?
Yeah.
A complicated organism. So.
Yeah, so this is something that has been brought up in the past. I've actually talked to Josh Vernovitz about this, and at the time I thought, well, you can't really do it because you're not in a steady state, right? Because you're only giving them an hour to metabolize, you don't know that they're there, but he says that you can do it in theory with some of the tools. So it's something we've thought about, but we haven't really pursued.
Can you just go on to slide number 60?
60? 60?
60, yeah. So we had that heat map with.
Mm-hmm.
Yeah, so after, so you're feeding till 4 hours constant, and then you see M plus N peaks, which are upregulated, and N plus 0 peaks are downregulated or present less. So
Yes.
it's getting labeled really fast.
Well, within an hour. So what we do is we give them an hour to metabolize. So each of these time points is when we start the experiment, and then we give them 1 hour to, you know, we inject, give them an hour to metabolize, and then we collect. Yeah,
That's why I was like,
Yeah.
how can the labeling be at 0?
yeah,
Yeah.
yeah. But you know, if you inject at time 0 versus time 12, you get a very different pattern, right? Which still blows my mind. You know, it's really interesting.
Yeah. Thank you.
Mm-hmm.
The about oxygen and carbon dioxide, that's very interesting.
Mm-hmm.
And I can imagine in USDA, I've seen they measure people in a chamber
Yeah,
and to measure that same metabolism.
yeah.
They also see the RQ being higher than 1, and how do they relate, like explain it in human?
Good question.
Glucogenesis?
Yeah, it's a good question. I haven't followed the mammalian literature, and I've just cursorily looked at papers, and I haven't seen anything greater than 1 in mammalian
Oh, okay.
work. Yeah, so the only place we've seen it is in our work and in Alex Keene's work. Yeah.
How did you measure myeloid-CoA? The CoA, how did you measure that?
To measure using DESI.
But is it like easy to degrade, hard to detect? Tricky for you?
So
Myeloid-CoA?
I should you know, introduce the caveat, which I should have done at the beginning, that all of the DESI measurements are M+1, or so parents, right? They're not. There's no MSMS for most of that. So, you know, I'm convinced that that's what it is because there's a series of CoAs that line up, you know, with the right specificity, but we haven't confirmed it.
Isn't it the MRM that has MS2?
Pardon me?
Isn't like the MRM with certain number of metabolites on the slide that's basically it goes MS2? That's not DESI experiment?
So we've done MRMs in DESI for a few things, but I don't think in my talk today there was any of that. So we've done it mainly with drug metabolites and looking at, you know, drug distribution in cells. Yeah. Yeah, the problem with the DESI, at least the way it's set up in the Waters instrument, is that you can only do 1 measurement at a time, right, for the MRMs. So really all we can do is like 1 MRM image.
No, I wasn't talking about imaging. It's like the overall metabolites you put in green saying that you can measure. I think summary slide say
Yeah.
pyruvate you can measure, serine you can measure, and myeloid-CoA is part of it.
Yeah,
It's
yeah,
not
yeah.
necessarily imaging experiment.
Yeah, no, you're right, you're right. So some of the work that we did do for the fly work was extracting the flies and then spotting them onto DESI plates, right? So it's more of a high throughput assay, again, using the parent ion. And so it could be that that's where that data came from, was still from DESI, yeah. I would have to go back and look at, because I don't think that our main list has, because we have kind of this targeted method, right? The HELIC method, it's ion switching that we use all the time. And I can't remember if that has myeloid-CoA, I'll have to confirm.
So given human, in addition to they don't sleep well, they also have higher locomotor activities.
Mm-hmm.
So in your opinion, how much of the locomotor activity or how much of the exercise we do would influence the entire metabolism?
A lot, actually, a lot. I mean, so there's a lot of factors. We have a number of other studies, human studies, where we're looking at actigraphy and how that relates to human metabolism. And what we find in certain populations, and I don't know if this translates across populations, so in the case I'm going to talk about is individuals with sleep apnea, which tend to be a little bit with higher BMIs, right? So they have higher BMIs, typically in the overweight category. And what we find is that activity at certain times of day predicts changes in metabolism and predicts changes in sleep quality, right? And so I think that activity actually has a substantial impact, and the timing of that activity matters, right? So whether you are active in the morning or the afternoon, it changes the way that then impinges on things like sleep and then metabolism overall. Yeah.
So the glucose metabolisms in the fermi mutant is abnormal compared to the wild type, right? The last one.
Yes. Yeah.
So, but like, I think it's maybe related to his question, but fermi mutant is active.
Yes.
More active compared to wild type.
Yes.
So I think it's like glucose metabolism. Like it's kind of like I don't lower, I mean, delay for your experiment. So I wonder, like, why it is like that? Because like Fumi needs more energy because they have to
Yeah.
activate more.
Yeah.
So I assume they have to use more glucose to active.
So I think that what we're seeing, let's say if we look at this data, right, is that and if you think back to the activity data, right, so the fumin is very active during the night as compared to either the sleepless or the wild type, right? They don't have that sort of drop in activity. And so what we're seeing here is at ZT0, we're seeing that sort of biosynthetic increase, which would be the end of that activity period for fumin, right? And so I think that it's just a reprogramming of the way that they're taking in, or that they're able to process this glucose when it's given as a challenge. And I think that that's the important thing to remember also, right? Is that experiment here, it's a challenge experiment. So you're now pushing the physiology a little bit, as opposed to just a steady state measurement where you're not actually challenging them, right? And so I think that that's what this is showing, is that because of this, differential behavior, that we're seeing a different way of processing, which shifts that a little bit, right? And so I think that there may be a shift due to the behavior, but I don't think that shift is due to, let's say, feeding, right? And that's what our feeding assay shows. Yeah.
So you think it's maybe reprogramming is maybe happening during the night?
Yeah, I think it's a response to the activity, right? I mean, if you need energy at night, then you're going to have changes in all these metabolites and these pathways, right? And I think that has to, at some point, control the way that all the other parts of the machine work. And so to me, I definitely think that there's a relationship there. Yeah. Good question.
I have one question.
Sure.
This might be kind of out there. So, in the fly field, a lot of people study post-meal sleep, right? I think even humans, if we don't have to go to work, sometimes I guess we'll take a nap in the middle of the afternoon after lunch. Like some countries, they do it, right?
Yeah.
Do you think that that sleep is also changing metabolism and how we might be actually metabolizing our lunch? Or is there some kind of bidirectional relationship between the two? And the fact that we're not taking a nap after lunch in humans,
Right.
is that changing the way that we might
Interesting
we should
question.
be
Yeah.
metabolizing food?
Yeah. So if I'm going to speak outside of school because I'm not a fly biologist, but so flies have the siesta, right? So after afternoon siesta, they'll like take a nap, and then they'll show that activity again, as I was showing in that activity profile. And so as humans, if we're not doing that, are we kind of losing out on some positive benefit?
Yes.
I mean, so it has been shown very clearly that taking a nap in the afternoon improves all sorts of function, right? Cognitive function, but short naps, right? Like so they're talking 20 minutes or half an hour or something. And so I've seen studies showing that that's a really important, or can be an important way of, improving function. So I would say that there's evidence that napping is important. Whether that's related to the postprandial, phenomena or not, I don't know. But certainly from a neurocognitive perspective, yeah, I think that, we should be encouraging everybody to take a nap.
Right. And we don't know whether that type of sleep how different is that type of sleep to actual nighttime sleep?
Right. Yeah, and I think that one of the reasons why they suggest a shorter period is because if you fall into a deeper sleep, then it actually has a negative effect later on. Yeah.
I suppose, kind of conversely, if you were, a lot of people also tend to have like late dinners or like some sort of late night food before they go to bed.
Mhm.
Would you suspect that the active digestion during the main hours of sleep might be disruptive to the
Yeah.
rhythm here in some way?
Good question. So there's been a fair amount of work done on this. Folks like Frank Shear at Harvard have looked at timing of food intake. And the evidence shows in terms of things like weight loss, for example, is that if you take two groups and they're on the isocaloric diet, so they're eating the same thing, but one group is eating a couple hours earlier through the day than the other group, that the rate of weight loss is much, and it's pretty dramatic, much different for the group that's eating earlier. They lose a lot more weight than those that take in calories later in the day. So I think that, yeah, that's definitely an important consideration, and those kinds of effects are becoming increasingly appreciated for sure. Yeah.
Any more questions? If not, please join me in thanking Dr.
Thank you. And thanks to Oliver, Jeanette, and Joanna for inviting me, and wonderful hospitality. Thank you.